In this video, we're going to move on to question number 2, which focuses on a genetic trait, a couple of genetic traits in mouse. So we have a lot of information to keep track of here in the lead into this question, so we need to read carefully and make sure that we're understanding the significance of everything that we're reading here. So this question pertains to a peptide hormone, a protein hormone called leptin, which regulates energy balance and hunger and satiety, among other functions. Now, in mouse, this protein is encoded by a gene called LEP, L-E-P. And mice that are homozygous for a mutant allele of this gene, LEP-OB, so the name of the gene is LEP, and the mutant allele at this gene has a superscript along with the gene name that indicates something about the mutation and the effect that this mutation has. Now, in the case of this allele, LEP-OB, the effect of that mutation is that it causes disruption in energy regulation and metabolism, causes weight gain. It's associated with diabetes, right? And you can see an image of a mouse over here on the right that is LEP-OB, LEP-OB, so they are homozygous for that mutant allele, and you can see they're much bigger than we'd expect an average mouse to be, and these mice also end up being diabetic. So important information is coming though, because it says heterozygous mice appear wild type. Heterozygous mice appear wild type. So what that tells us is that if we have a mouse that has one allele, that's the normal wild type allele, and one allele of this OB type, these mice are perfectly fine. These are wild type mice. So that's important information. Let's file that away and continue on with what other information we have in this problem. So now we're looking at a separate gene, gene B, that determines mouse coat color. And there's a dominant capital B allele that encodes black pigment and a recessive lowercase b allele that encodes brown pigment. Now, the question tells us that if we cross two true breeding strains of mice, with parent strain 1 being LEP-OB, LEP-OB, and lowercase b, and parent strain 2, who is LEP-LEP, so two wild type alleles at the LEP gene, and capital B, the offspring of that cross are all wild type with regard to their metabolism, so they have normal weight, they're non-diabetic, and they have black fur. So just like we talked about for question number 1, the progeny of the cross of two true breeding strains is going to give us important information about the dominance relationship between our alleles at these two genes, right? And so we need to think carefully about what this information is telling us. So again, with regard to the LEP gene, we already said that heterozygous mice, so mice that have one of the wild type alleles and one of the mutant alleles, ends up being wild type. And so what that tells us is that the wild type phenotype with regard to the LEP gene is dominant. And remember, that phenotype was associated with that wild type LEP allele. The increase in weight and diabetic phenotype is recessive. Right? When we have a mouse that has two of those LEP-OB, LEP-OB alleles, we end up with a mouse that has higher weight and is diabetic, right? If we have animals that have even one of this wild type LEP allele, right, heterozygous mice appear wild type, those animals do not show the disruption in metabolism and the weight gain. All right, so that's going to help us figure out our phenotypes of our parent strains 1 and 2. Now with regard to our B gene, we're told that this allele, capital B, encoding the black pigment is dominant. We don't have to even figure it out, right? So recessive B allele encoding the brown pigment, we know the dominance relationship here, right? So black fur is dominant over brown, and the black fur allele is the capital B. So given this information, we can now look at our parent strain. So we had parent strain 1, who was LEP-OB, LEP-OB, and lowercase b, lowercase b. And then we have parent strain 2, who was LEP, LEP, and capital B, capital B. So with regard to the leptin gene, parent strain 1 is going to have a higher weight and be diabetic. And with 2 recessive alleles at the B gene, parent strain 1 is going to have brown fur. Parent strain 2, having 2 wild type alleles at the LEP gene, is going to have normal metabolism. So normal weight, non-diabetic, and having 2 dominant alleles at the B gene, black fur. Right, so we have figured out the phenotypes, the physical expression of the alleles at these 2 genes, the LEP gene and the B gene, for parent strains 1 and 2. So let's move on to question number B then. Question number B asks, what would be the genotypes of the F1 mice generated by the cross of these parent strains? So if we look at parent number 1 is LEP-OB, LEP-OB, lowercase b, lowercase b. Parent number 2 is LEP-LEP, and uppercase B, uppercase B. So homozygous recessive at both genes for parent 1, homozygous dominant at both genes for parent 2. And if we think about the cross between these 2 parents, we can see that parent number 1 only has one version of the leptin gene to pass on, being true breeding. Right? So parent number 1, 100% of the gametes from parent number 1 are going to contain that LEP-OB allele and an allele at the B gene, which is the recessive allele. So 100% of the gametes are going to look like this, right? For parent number 2, 100% of the gametes are going to have the wild type allele at the leptin gene and the dominant allele at the B gene. 100% of the gametes. This parent only has the wild type allele to pass on at the leptin gene, only has the dominant allele to pass on at the B gene. And so if we combine these gamete types, so this would be the process of fertilization, right? These animals would mate. We would see gametes fertilizing. The offspring, F1 offspring of this cross, would get one allele from each parent at the leptin gene, right? A wild type allele from this parent, a mutant allele from this parent, and one allele of each type at the B gene, right? And we call this state of having different alleles at these genes, these F1 animals, would be heterozygous for both of these genes, the leptin gene and the B gene. So if we answered the question, what would be the genotypes of the F1 mice generated by the cross of these parent strains? Yep, they would be LEP, LEP-OB, capital B, lowercase b, heterozygous at both genes. Now let's go ahead and turn our attention to questions C and D. And I'm going to scroll down here and give us some more room. So question C asks, if these F1 mice are intercrossed, so crossed with each other, what proportion of the offspring would be expected to be non-diabetic and black-furred? What genotype is associated with that phenotype? And then question D asks, what percentage would be expected to be diabetic and brown, and what fraction would be expected to be non-diabetic and brown? What about diabetic and black? So these two questions are kind of asking the same thing, right? They're asking, if we take these F1 mice and we intercross them with one another, what offspring proportions in terms of phenotype and genotype do we expect to see, right? But for most of these two questions, we're asked about phenotype. And so there are a couple of different ways that we can tackle a question like this. So I'm just going to write once again, so we don't lose track of this, what the genotypes of our F1 mice were. So they were heterozygous at both of these genes. And we're intercrossing, so we're taking an F1 mouse, crossing it with another F1 mouse that has the same genotype. Now, a couple of ways we can do this. We can do this the long and time-consuming way, which is to create a Punnett square that shows all of the possible offspring that could be produced given random combinations of all of the possible gametes that these F1 animals could make, right? So gametes, we remember, are produced through the process of meiosis. So I'm going to show you this long and tedious way first, and then we'll go over a quicker and easier way. But let's do the long and tedious way first. So meiosis is going to produce gametes. Remember that gametes, reproductive cells, are haploid. So whereas our animals that we start with are diploid, having two of each allele, our gametes are going to be haploid and have only one allele at each gene. So one allele at the leptin gene and only one allele at the B gene, right? And so easy way to figure out what the genotypes of these gametes is going to be would be to put our two possibilities for the leptin gene, so our two alleles that this F1 could pass on, and then those are going to combine with the two possibilities at the B gene, because every gene has to be represented, but represented only once in each gamete. And so then our gametes have the following genotypes: wild type LEP, dominant B, wild type LEP, recessive B, LEP OB, dominant B, LEP OB, recessive B. All right, so these are our gametes. Notice that they only have one copy of the leptin gene, one copy of the B gene. They are haploid. So we're crossing the F1s with each other. We could create a Punnett square with our gamete types for our two F1 parents down the left side and across the top. I'm just drawing circles around these to indicate that we're looking at gametes. Maybe these are spermatozoa over here. And then the ova would be down the other side. These are gametes combining in all possible combinations to give us the next generation of animals. Now, we're starting to see why this is not the favored approach for this, because it's going to start to get kind of crazy to keep track of all of these alleles and not get mixed up, right? But we could do this. So LEP, capital B, capital B, LEP, capital B, lowercase b, lep OB, capital B, lep OB, capital B, lowercase b, so we have to fill in all of these, making sure we don't make any mistakes. Lowercase b, lep OB, capital B, lowercase b, lep OB, whoops, and there we go. That's our first mistake. Should be lowercase b, lep OB, capital B, lep OB, capital B, lowercase b, lep OB, capital B, lep OB, capital B, lowercase b, lep OB, capital B, lowercase b, lep OB, lowercase, lep OB, we're coming down the home stretch, capital B, lowercase b, and lep OB, lowercase b. Now that was time consuming, and as you can see, it's super easy to make mistakes in trying to combine these gametes in all possible combinations. So I don't recommend doing this again. I'm going to show you a quicker and easier way to determine the probabilities of any particular offspring showing up in the next generation. But let's go ahead and go with this. Let's say you did decide to do it this way. Could you answer the question like this? Absolutely, you could do this. So again, the question C asked us, if these F1 mice are intercrossed, what proportion of the offspring would be expected to be non-diabetic and black? So we have to remind ourselves of the dominance relationship for our two genes and the alleles at these two genes. So for the leptin gene, remember that being non-diabetic was actually the dominant trait, as was black fur. These are both the dominant trait, right? And so if you have even one of the lep allele, so lep and then anything, it doesn't matter what the other allele is, you're going to have this non-diabetic dominant phenotype if you're a mouse. Likewise for the fur color gene, because the black allele is dominant, having even one of that dominant allele, doesn't matter what the other allele is going to give black fur. So what we can do then is look through our table and find all of the genotype combinations that give us at least one of the wild type leptin gene allele and at least one of the dominant B allele. And so if we do that, we would say there's one, there's one, there's one, there's one, there's one. This one is not one because it doesn't have at least one dominant allele at the B gene. Here's one. This doesn't count. It doesn't have at least one dominant allele at the B gene. Here's one. Here's one. This doesn't count because it doesn't have at least one dominant allele at the leptin gene. Likewise here, that doesn't count. Here's one. This doesn't count because it doesn't have a dominant B. This doesn't count because it doesn't have a dominant leptin allele. And if we go through here and we count up all of our check marks, we should have 1, 2, 3, 4, 5, 6, 7, 8, 9 out of our 16 that have that dominant phenotype pattern. And if this is starting to sound familiar to you, it's because we talked about this in the lecture video for doing genetics problems that are dihybrid crosses, where we're looking at inheritance at 2 genes and both parents are heterozygous at both genes. We expect to get that 9 to 3 to 3 to 1 ratio, where 9/16 we expect to have the dominant phenotype at both, 3/16 we expect to have the dominant phenotype at one, recessive at the other, 3/16 we expect to have the reverse, recessive at the first and then dominant at the other, and then 1/16 we expect to be recessive for both traits under investigation here. So that 9 to 3 to 3 to 1 ratio is what we expect, and we could get that if we counted up for these other traits here. What's the likelihood of being non-diabetic and brown? Non-diabetic being the dominant trait, brown being a recessive trait. Here we're looking for animals that have at least one of the dominant leptin gene, but their genotype at the B gene would have to be homozygous recessive. And so if we go through and we count up all of those guys, we should come up with 3 out of 16. We can do that really quick. Let's switch colors here. We'll make this one red. So let's find everything that has little b, but at least one of the dominant alleles. So let's go row by row here. Here's one. It's got dominant alleles and little b. Here's another one, little b, at least one dominant allele. And then we are looking for little b, and at least one dominant allele. Here we go. And that's it, 3 out of 16. All right. So we could do this for all of the other phenotypes that are being asked for here. And as we can see, we get that 9 to 3 to 3 to 1 ratio because we are dealing with parents in this cross, both of which are heterozygous at 2 different genes, right? So that is one way that we could solve this problem. As I said, though, I don't think that's the best way to solve this problem. I think that it's better to actually use probability and maybe some small 2x2 Punnett squares to do this instead. So let me give myself some more space to work here. All right, so let's do this a different way. So let's do this with probability. So if the F1 mice are intercrossed, what proportion of the offspring would be expected to be non-diabetic and black, and what genotype is associated with that phenotype? That would actually be our starting point if we wanted to do this using that quicker method and probability instead. So again, our F1s, let's just write our genotype so we don't forget. They are heterozygous at the leptin gene, one wild type allele, one mutant allele, and heterozygous at the B gene. So both of the F1s that we're crossing against each other are going to have the same genotype. And so if we think about this, crossing F1 that's a heterozygous animal with another F1 that's a heterozygous animal, we could actually think about this and capture this kind of in a snapshot in our mind as all possible offspring being the product of 2 smaller Punnett squares, right, which is a little bit easier to visualize, right? We'll do the LEP Punnett square over here. So both of these F1s are going to be passing on one or the other of the leptin allele and one or the other of the B gene alleles, either dominant or recessive. And so this is a little bit easier to keep track of. LEP-OB, LEP-OB. We can see that this is just the same pattern of homozygous dominant, heterozygous, homozygous recessive in 1 to 2 to 1 ratio that we expect to see with this kind of heterozygous cross, right? Likewise over here, 1 out of 4 homozygous dominant, 2 out of 4 heterozygous, right? And so we can think about this cross and break down this cross as sort of smaller Punnett square crosses where we're just focusing on one gene at a time and thinking about the probabilities of ending up with different offspring, one gene at a time, and then combining those probabilities together to account for all genes of interest here. So to account for our leptin gene and our coat color gene. So how would we do that? So let's go to question C first. What proportion of the offspring would be expected to be non-diabetic and black? So non-diabetic being the dominant phenotype and black fur being the dominant phenotype. 3 out of 4 we would expect to show the dominant non-diabetic normal weight phenotype, and 1 out of 4 to show the diabetic and higher weight phenotype, right? Just thinking about the leptin gene. Over here at the coat color gene, likewise, we expect 3 out of 4 to have black fur, 1 out of 4 to have that recessive trait of brown fur. So 3 out of 4 having at least one of the dominant allele is going to give black fur, and then only this homozygous recessive genotype giving us brown fur. So what is the genotype that would be associated with the phenotype of being non-diabetic and black? Non-diabetic and black would be having even one of that non-diabetic allele, and anything else, doesn't matter what the other allele is, and even one of that black coat color, right? Doesn't matter what the other allele is. It could be homozygous dominant or heterozygous, doesn't matter. And so what's the likelihood of having at least one of this wild type leptin allele? Well, that's 3 out of 4. We just went through that, right? At least one would be 3 out of 4. At least one of the dominant allele at the B gene, likewise, 3 out of 4. And because we want this genotype, at least one dominant allele at the leptin gene, and this genotype, at least one dominant allele at the coat color gene, in the same animal, we want these two things together. This is an and. We're going to multiply the individual probabilities, and we would come up with 9/16, which is exactly what we got from that giant, ugly Punnett square, right? What percentage would be expected to be diabetic and brown? So diabetic is the recessive trait, and the genotype that would give us diabetic mice would be homozygous recessive at the leptin gene, and brown also being the recessive trait, we would have to have lowercase b at the B gene. And if we look at our small Punnett squares, what is the likelihood of having homozygous recessive genotype at the leptin gene? Well, that's right here. Homozygous recessive is our 1 out of 4, right? So 1 out of 4. Likewise, homozygous recessive at the B gene, 1 out of 4. And because we want these two gene allele combinations in the same animal, this is an and, we want lep-OB, lep-OB, and little b, we are going to multiply these individual probabilities, and we come up with 1 out of 16, right? What else were we asked for here? That was diabetic and black, non-diabetic and brown. What about diabetic and brown? Well, diabetic is going to have to be homozygous recessive. Wait, that's what we just did. Memento. We don't want diabetic and brown. Non-diabetic and brown. Actually, we'll do diabetic and black. Diabetic and black fur. So diabetic being the recessive trait, black fur being the dominant trait. Likelihood of being diabetic, the recessive trait, that is 1 out of 4, right? Lepo B, Lepo B. Having black fur, that's the dominant trait, that's 3 out of 4. We want this and this, we're going to multiply, and our total probability is 3 out of 16. And then our other combination would be non-diabetic and brown. Same logic. This is our dominant trait, and this is our recessive trait. Non-diabetic, 3 out of 4 probability, at least one of that wild-type leptin allele. Brown recessive trait, brown recessive trait probability is going to be 1 out of 4. It's that homozygous recessive genotype. Because we want this and this in the same mouse, we're going to multiply 3 out of 16. As you can see, we get our 9 to 3 to 3 to 1 ratio. It's just a lot easier to keep track of small Punnett squares, and once you've got a little bit of practice with this, you won't even need to write the Punnett square out because you'll be so used to this 1 to 2 to 1 homozygous dominant, heterozygous, homozygous recessive genotype frequency and this 3 to 1 phenotype frequency that you can just picture it in your mind for each gene, and you won't actually have to write out the Punnett square to be able to visualize that. So this, I think, is an easier way to do that, but whatever way makes more sense to you is the way that you should use. And you should come up with exactly the same answer. So it's all about what works for you.